Hi, Miss Nikki here. Welcome to Chapter 4. Let's go through some of the problem issues that students have in this Chapter 4 material. First thing we have to understand is that the plasma membrane is going to serve as a boundary. So it says it separates the internal environment from its external environment, and that way it can regulate what comes in and out of a molecule. So we know we have our cell, and then around our cell we have that phospholipid bilayer. I'm not going to draw all the little phospholipids, right? But that phospholipid bilayer decides what can get in and out of the cell. And to maintain a normal or steady internal environment for this cell, so the inside stays stable, we call that homeostasis. And we're going to need to regulate what can get in and what can get out. So if you think just basics, remember we need glucose plus oxygen in order to generate energy, or ATP. So we have to be able to get glucose and oxygen inside the cell, where the cells have mitochondria, which actually take the products of the breakdown of glucose with oxygen and turn it into ATP energy for the cell to do work. Here's our image of our phospholipid bilayer, and then we see that our proteins are embedded in this phospholipid bilayer or plasma membrane, and then we have these glucose tags. We should have talked about glucose already. I don't remember which chapter it is in because we've switched books, but we have carbohydrate tags. We're going to have proteins. Here we have lipids, right? And then how did we make the proteins? We made them from DNA. RNA, so these are nucleic acids. So we've covered all the 4 biomolecules before, right? Now let's look at individual proteins and what kind of function they can serve within the cell. The first type we have here is a channel protein. So if an ion, remember ion's something that's positive or negatively charged, sometimes we're going to have a protein that acts as a channel. These positive and negative charges have a hard time squeezing their way through this plasma membrane. Water can squeeze through, and so can non-charged. And we'll come back and talk about this in a second. But if you're a charged molecule, you have to have some help, and so you have to have a channel protein. Here we see a carrier protein. So glucose is a little bit larger than just an ion, right? What is glucose? 6 carbons, 12 hydrogens, 6 oxygens. So it's a little bit larger. I wish they had drawn it kind of like that. It would have made it seem like it was a little bit larger. So glucose has to have a carrier to get across the plasma membrane. And then over here we have a cell recognition protein. So this is a glucose tag that's sitting on a protein. So they call it a glycoprotein. So these glycoproteins are different in every person, and this is where we come up with this term. You're looking for somebody that's a match if you want to do an organ transplant. These are called major histocompatibility complexes or factors. So everybody's branching is different. It's how your immune system knows which cells belong there and which ones are foreign invaders that need to be digested. Next, we have receptor proteins. This is how a lot of hormones work. So hormones will bind to a receptor, and then they'll cause action to happen inside the cell. So the entire hormone doesn't come into the cell, it just docks or binds to the protein and allows for this kind of domino effect or cascade effect for action to happen inside the cell. And then here we have enzymes. So some proteins are enzymes that have specific jobs. They catalyze specific reactions, cause the reaction to happen. So you can see this item binds and then it changes or breaks apart into pieces. This is probably not the best picture. We have some more coming up that are a little bit better. So this lends to the whole idea that the plasma membrane is selectively permeable. So the plasma membrane regulates what goes in and out of the cell. So which molecules can freely cross and which ones require some sort of carrier. It depends on the size and then the charge of the molecule. I love this image here. It kind of puts everything in picture form, all the stuff that we're getting ready to talk about. So here is non-charged. You can see the non-charged moves freely across the membrane. So this non-charged example, think of oxygen, carbon dioxide. This is the process that we're going to call diffusion. That's when a non-charged molecule moves from high concentration to low concentration. Then we see water here moving. Water moves through a carrier. Sometimes it can also get through the plasma membrane, but it does have these things, they're called aquaporins. It's not super important right now. But water moving, this is osmosis. Water is moving from high to low concentration, but it's based more on the solutes. So water is always going to where the solute is highest. And then, so the example would be water. So both of these, we're going to learn, both of these processes are called passive. There's no ATP required. Now, if I have a charged molecule, in order for it to get inside the cell, this is active, so active transport, so ATP is required, and you need a protein. So you'll hear me call it protein-assisted active transport. This is for charged. So what would be charged? Sodium, potassium, the chloride. And then if you have a really large molecule, this is also active transport. So this one, you're going to actually see the plasma membrane come around, engulf, and bring the large molecule in. You can imagine if you just cut a hole in the plasma membrane to bring this really large item in, you'd have a problem, right? So sometimes I'll call this membrane assisted. I've also heard it called vesicular assisted, but you'll probably hear me use protein assisted and membrane assisted. That's how I learned it. It's kind of stuck in my head. So we're going to go through each of these in a little more. Here's another great table for you. Look through this table. Here's diffusion, no energy required, so we would call that passive. Towards the lower concentration, that means items move from high to low. And example, this says lipid-soluble molecules and gases. We're kind of going to focus on the gases. But also anything that's a lipid can pass through the phospholipid membrane. So let's go through each of these, but it's always good to come back and look at this table. This first example is diffusion. So if I had dropped a dye, crystallized dye inside this fishbowl, what's going to happen? Eventually the dye is going to spread out and you're going to have an equal distribution of water molecules and dye crystals, right? Or dye molecules, whatever you want to call them. The same thing happens if someone's smoking. So if someone's smoking at the front of the room, I guess now the cool thing is vaping, right? If someone's smoking or vaping up front in, say, a classroom or up front in a restaurant, it eventually makes its way to the back, right? This is diffusion. These are non-charged molecules moving from high to low. Do we see how this is a high concentration and it's moving to where there's a low concentration? And there is no ATP required. So a great example of diffusion is oxygen and carbon dioxide. I've said this a few times. So we're trying to get oxygen in, right? The whole point is we need this to make ATP. So oxygen goes down the trachea, the bronchus, the bronchioles, and goes all the way out to the edges where this sac is located. But then how do we get the oxygen from the tube system into our blood vessels? Remember, the oxygen has to get into the blood and then it has to bind to the red blood cell. So this happens by diffusion. There's no ATP involved. It just transfers over from wherever it's high to wherever the oxygen level is low. So next is osmosis. So with this red line, they're trying to show you a selective permeable membrane. So this would be just like our plasma membrane, right? The solutes are the green little spheres, and then the water is the blue. So count up the number of green. You can see, take a guess, I'm not going to, like, say 20 green, right? And then on this side, there's 2, 4, 6, and let's say 8 solute. So do you see how the solute is higher over here and it's lower over here? Well, the solute can't get through the plasma membrane, but the water can move. So the water moves towards the higher solute concentration. It's trying to even out the concentrations. So if my solute is high on this side, which way do you think the water is going to move? Right. So the water is moving toward where the solute is the highest. But just look at the water. Do you see how the water is high on this side? Count all the blue spheres. Is the water still moving from high to low? Yes. Is water moving from high to low to where the solute is highest in order to even out the concentrations? Yes. All right, so let's look at the next slide for a real life example, not this apparatus example. So now we're going to move to a practical application. Here's a red blood cell, right? This is going to carry oxygen. So if you have an isotonic solution, that means the inside and outside is equal. So that means we're saying the water out here and the water in here is equal. That's also kind of saying the solute is equal, right? Or relatively equal. Because where does water like to move? Water likes to go to wherever the solute is high. All right, so this example, I'm going to draw a little box and I'm going to draw some water because I hate it that they don't tell you or explain to you that this is a solution. They just show you an image. So here's your cell and I'm telling you it's in a solution of water. So it's in a hypotonic. So hypotonic means low solute in the solution. So if the solute is low outside, the solute inside is high compared to the solute that's outside. So where does all the water go? The water rushes in to wherever the solute is highest. So what's going to happen to your red blood cell? If all the water is rushing in, it's going to expand and expand, and eventually it can lyse. That would be bad for us, right? It's carrying oxygen to our tissues so that they can break down glucose and make ATP. So here, same example, except for the opposite, right? Here's a hypertonic. So now you have high solute here compared to what? Low solute on the inside. So where does all the water that was inside the cell go? It goes out to where the solute is highest, and it causes the red blood cell to shrivel. Again, not working at its optimal level, right? It's going to have a hard time carrying oxygen. It may not be able to work at all. With plants, they have a little bit more protection because they have the cell wall. So the cell wall is not going to allow for the bursting or the shriveling that we would see with the red blood cell. So this cell wall acts as an added protection. Remember, it's made of cellulose. It's a little bit stronger. And then here we can see that the plasma membrane is pulling away from the cell wall, but the cell wall is still intact. Here we have facilitated transport. We still have no energy required, but you're going to have a protein that's going to help. So the carrier protein is going to allow for certain molecules to travel down their concentration gradient from high to low, and no energy is required. Usually the protein will undergo some sort of shape change, but there's still no energy required. We still kind of call this passive transport. They just need a little help. So this is our first example of active transport. The other two, those were passive, no ATP. So active transport, we're moving from low to high concentrations, and ATP is required. This would be an example of protein assisted. So you're going to have a protein. We change colors. You're going to have a protein here, and you're going to take these ions. So this is how you move ions or charged molecules. And you're moving them against the current from low to high. Sometimes I say this is like swimming upstream. You've got to put a lot of energy in to go against the current. So here we have these 4 sodiums, and we're trying to move them outside where there's 5 sodiums. So we're moving them against the current. We're going to have this sodium-potassium pump. So there's a few more pictures. Let's look at those. Here we see ATP binding the protein. It breaks off one of the phosphates, and now we have ADP. So where did that energy go? That energy went into this protein in order for the protein to be able to change shape. So you're going to see the sodiums are bound here. And now we see that the protein has changed shape. Remember, it was this way. Now it's opened up to the exterior and it's dumped the sodiums off. So we went from low to high. We used ATP and we used a protein. So this is active transport, or you can call it protein assisted active transport. What happens next is it picks up potassiums. The potassiums bind, and now this phosphate is ejected off of the protein, and now the protein opens up and drops the potassiums. So what are we doing? Big picture, we're moving sodiums this way and we're moving potassiums this way. This is critical for muscle action. When I say muscle action, I'm talking about heart, I'm talking about blood vessels, any place you have muscle, not just your skeletal. Okay, this is another example of active transport, and this is called membrane assisted. This requires ATP, and you're moving from a low concentration to a high concentration. So this is the cell. Let's say the cell made a protein, and I want to get that protein outside of the cell. Maybe it's a protein that's going to stick and allow another cell to bind to this cell. So we'll talk about those extracellular proteins in just a second. So we have gone the pathway, right? DNA to RNA. We've made a protein. We went through the Golgi apparatus. We packaged the protein, and now we're sending the protein to be excreted. You see how this is called a vesicle? This is a vesicle right here. It's a phospholipid bilayer, but it's a tiny little sac, right? That vesicle is going to merge with the plasma membrane and allow you to remove the protein that you made or move the protein from the inside to the outside. So again, this is active transport. It requires ATP to do this action, and you're moving from where you have a low concentration to a high concentration. You're moving against the current, and you need a membrane to help you get there. So here we have another example. This is called phagocytosis. This is a type of endocytosis. Cytosis. There we go. So this is just an example. This is when you bring in a solid. And endo means in, right? Into cell. So here we have a solid and we want to get it inside the cell. You see how the vesicle just kind of invaginates and engulfs the solid particle. So this could happen, lysosomes do this. Lysosomes will engulf particles, chew them up, spit them out, right? Sometimes we're going to do this with hormones. Sometimes we could do this with neurotransmitters. I couldn't get the word out. So these are the chemicals that help neurons work, right? That signal neurons. So chemical signals. Pinocytosis is another example of endocytosis, except for this time we're taking in a liquid. So this is some sort of liquid that's being brought into a vesicle or transported inside the cell. There's also receptor-mediated endocytosis. So you can see these are receptors involved with binding a solute, and then that triggers this invagination or vesicle formation inside the cell. So basically with endocytosis, you have this one, receptor-mediated, then you have phagocytosis, then you have pinocytosis. So all three of these are examples of endocytosis. One of the last few things I want to mention here are proteins, right? They're anchored inside the plasma membrane. And if we're inside the cell, so this is intracellular, and this is the extracellular. So remember, we have our DNA copies itself to RNA, which is then going to make a protein. And then that protein can be shipped outside the cell. We just saw exocytosis. So we do have proteins outside of a cell that allow cells to stick together and form sheets of cells, which we're going to call tissues. So I just want you to realize that if there are proteins outside of the cell, who made these proteins, right? They came from inside the cell. They came from the code, the DNA code inside the cell. So if we want to create junctions between cells, we're going to have different types of junctions depending on what function we want. So this one, adhesion junctions, you can see this is the plasma membrane. Here's cell 1 over here, cell 1 over here, and here's cell 2 over here. We want to connect them, but we still want them to be flexible. So this is an adhesion junction. Probably a good example would be your skin. They're together, the cells are together, but they're still flexible. Here we have tight junctions. Tight junctions, it's not all the way tight, it's just tight at specific sections. This is if we don't want any spillover. So probably best example is the stomach. So we want, I'm going to say no spillover. That's probably not very scientific of me, but we do not want stomach acid to leak out into our abdominal cavity, right? So the cells of the stomach that are producing hydrochloric acid, we don't want that to be able to get out even into the stomach lining other than when we have food, right? When we put food in our stomach, then we want them to release their hydrochloric acid. But when we don't have food in our stomach, we don't want them to release it because then it would be destroying the tissue or the lining of our stomach. So now we have gap junctions. Best example for this is heart. And this we want communication and kind of synchronized communication. So we have a tiny little channel so that this cell and this cell can talk to each other. So in the example of the heart, you want your heart to contract all at one time. Your heart has to pump blood up and out against gravity. It has to all be forcefully contracting at one time. So that covers my summary for Chapter 4. If you have any questions, please just make sure you email me and let me know.